Welcome to Pharmacology Outline Channel. Today, we're exploring G-protein-coupled receptors, a critical component in cellular signaling and drug discovery. G-protein-coupled receptors, commonly known as GPCRs, represent one of the largest and most diverse families of membrane receptors in eukaryotic cells. These receptors are characterized by their 7 transmembrane alpha helices, which create a serpentine structure that spans the cell membrane. The extracellular domain is responsible for recognizing and binding to specific ligands, such as hormones, neurotransmitters, or even sensory stimuli like light or taste. When a ligand binds to the extracellular domain, it induces a conformational change in the receptor, which activates an associated intracellular G protein. There are several different types of G proteins, each with a different job. Some of the main ones you'll hear about are GS, GI, and GQ. G proteins are heterotrimeric molecules composed of 3 subunits: alpha, beta, and gamma. In the inactive state, the alpha subunit is bound to guanosine diphosphate. Upon activation by the G protein-coupled receptors, the GDP bound to the alpha subunit is exchanged for guanosine triphosphate. This exchange triggers the dissociation of the G protein into 2 active components, the GTP-bound alpha subunit and the beta-gamma dimer. Both components can then interact with and regulate various downstream effectors, such as adenylate cyclase or phospholipase C. The GTP bound to the alpha subunit is eventually hydrolyzed back to GDP, which deactivates the G protein. The subunits recombine, and the G protein-coupled receptors returns to its inactive state, ready to be activated again by another ligand. Let's take a closer look at the downstream signaling pathways activated by G protein-coupled receptors. GS stimulates adenylyl cyclase, an enzyme that converts ATP into cyclic AMP. Cyclic AMP acts as a secondary messenger, activating protein kinase A, which then phosphorylates target proteins to elicit specific cellular responses. This pathway is crucial in regulating metabolic processes, heart rate, and muscle contraction. On the other hand, GI inhibits adenylyl cyclase, decreasing cyclic AMP levels. Another significant pathway is the phospholipase C pathway. Activation of phospholipase C by GQ type leads to the hydrolysis of phosphatidylinositol 4,5-bisphosphate into 2 secondary messengers, inositol trisphosphate and diacylglycerol. Inositol trisphosphate stimulates the release of calcium ions from intracellular stores, while diacylglycerol activates protein kinase C. These secondary messengers play essential roles in processes such as cell growth, differentiation, and immune responses. The diversity and versatility of G protein-coupled receptors make them prime targets for drug discovery. For instance, beta blockers, which are used to manage hypertension and anxiety, work by blocking beta-adrenergic receptors, a subtype of G protein-coupled receptors. Similarly, antipsychotic drugs often target dopamine receptors, which are also G protein-coupled receptors. G protein-coupled receptors are also targeted in treatments for asthma, allergies, and depression. In conclusion, G protein-coupled receptors play an indispensable role in our body's ability to communicate and respond to various signals. By understanding how these receptors work, we gain insights into numerous physiological processes and how they affect our health and well-being. Thank you for watching. If you enjoyed the video, please like and subscribe for more pharmacology insights. Hit the bell to stay updated. Keep learning.